Hi everyone, my name is Tulio Campos. I'm a PhD student at the University of Melbourne. The title of my presentation is Predicting Gene Essentiality in Caenorhabditis elegans by Feature Engineering and Machine Learning. C. elegans is a free-living nematode that has been used extensively as an experimental model to understand a range of biological and molecular processes in multicellular organisms. The availability of a high-quality reference genome and the development of gene knockdown and knockout methods have underpinned numerous studies on gene function for this worm. Such studies have revealed genes that are essential for the survival of C. elegans and also genes that are non-essential. Other genes can be considered conditionally essential, depending, for example, on developmental stage, strain, or sex. However, this variability in essentiality can also be caused by technical bias in functional experiments, which is a confounding factor to define essentiality of such genes. Currently, there is a major demand for predictions of essential genes using computational methods. However, the features that define gene essentiality are still unknown. Therefore, the abundance of genomic-phenomic datasets available for C. elegans provides an ideal opportunity for an in-depth exploration of gene essentiality using large-scale feature engineering and machine learning. The methods used in the present study are illustrated in Figure 1. Here, we proposed and employed a scoring system to provisionally annotate essentiality for C. elegans genes using RNAi data available in worm-base. The results for this analysis are shown in Figure 2a. Then, we extracted or engineered features from those genes, derived from a diverse range of omics datasets. Using these genes provisionally annotated as essential or non-essential, and their corresponding features, we systematically trained and evaluated machine learning approaches for essential gene predictions. To this end, we prepared 3 datasets. The first contained all genes and their features. The second was a non-redundant set, which was created by removing features from highly similar genes at the amino acid level. And a third set contained the best predictive features found in common in the first 2 sets. For each of these feature sets, we created subsets ranging from 10 to 90% of the data. Each of these subsets were used for feature selection and training of 6 machine learning models with parameter optimization. We used the remaining 90 to 10% of the data for testing and evaluation using 2 metrics, ROC and precision recall curves. The final selection of strong predictors identified included features derived from single-cell RNA data, chromatin markers, as well as protein and nucleotide sequence-derived features. As shown in Figure 2b, the models consistently achieved area under the ROC curve values above 0.9 and area under the precision recall curve above 0.7. These results were confirmed using a bootstrap approach with the best performing machine learning method, namely random forest, gradient boosting, and extreme gradient boosting machines, as shown in Figure 2c. Using the final models, we predicted the essentiality of all genes used in this study. We then validated these predictions independently by using mutant allele data from the GExplore database. In this analysis, we found that predicted genes with higher essentiality probabilities were more likely to have alleles linked to lethal phenotypes than those with lower probabilities. And these results are shown in Figure 3. Next, we carried out 2 complementary analyses to gain more insights about essential gene location and function. First, we evaluated whether essential genes have a preferential location on chromosomes. As we can see in Figure 4, we found that essential genes were more likely to be found in or near the center of autosomal chromosomes. These locations correlated well with regions depleted of SNPs. Second, we performed gene ontology, tissue, and transcription enrichment analysis. This analysis suggests that essential genes are enriched in nucleotide or protein processing and are active in reproductive tissues. In addition, many essential genes are targets for small RNAs bound to the CSR1 protein, which is an argonaute known to protect genes in germline cells against transposable elements. This argonaute is also involved in chromosome segregation. In conclusion, we hypothesize that gene essentiality in C. elegans is likely defined by an interplay between epigenetic markers and small RNA pathways in germline cells or in the embryo, establishing a transcription memory profile that is maintained throughout development. Moreover, the machine learning approach employed here is promising for broader application in other species. We made the data and the code developed in this study available to the scientific community in a public Git repository. Finally, we propose that future work extending this research should focus on essential gene predictions in distantly related taxa. Thank you very much for listening.